Hi, my name is Pavithra Sundaravaradan, and I'm a microbiology sophomore at the University of Washington working with the Tate Bueno Lab on the effect of helminth infection and Notch signaling pathway manipulation on CD4 T cell response and gene expression. Here, we are studying helminth or parasitic worm infection and how it affects the regulation of immune responses and inflammation. Gastrointestinal helminth infection is one of the most common infections worldwide, affecting about 1.5 billion people. This project utilizes the parasite Trichuris mirus, a mouse model of human whipworm in the large intestine, to show that basophils, rare innate immune cells, accumulate at the site of infection and upregulate the expression of Notch receptors, part of a signaling pathway that regulates gene expression. Previous studies have shown that mice lacking basophil intrinsic functional Notch signaling had impaired worm clearance, causing faulty basophil localization in the intestine and decreased CD4 response. It is known that a strong immune response in the intestine is needed to expel these worms, and CD4 T cells are a key type of immune cells that is needed for this response. However, we still do not completely understand how this CD4 response is orchestrated. Basophils can interact with or impact these CD4 T cells and affect their function. Because of this, we are currently investigating the importance of Notch receptor expression on basophils for the anti-helminth immune response. To investigate how Notch signaling in basophils impacts CD4 T cell function during infection, we performed single-cell RNA sequencing on CD4 T cells from the intestines of uninfected and Trichuris mirus infected wild-type mice and mice without Notch signaling, specifically in the basophils. The single-cell RNA sequencing approach is informative as it is unbiased and can reveal lots of things about the Notch-programmed basophils and how they affect the CD4 T cell responses. This method provides us with information about the existence and behavior of different cell types in the large intestine and how they react to different experimental conditions. In this process, we take samples from the large intestines of the mice, isolate and sequence the cells, and then align the reads to a genome to produce read counts. After this, I am then able to use the read counts at R to compare gene expression profiles on a singular level and cluster them by differential expression as seen in the middle of the bottom row. After we cluster the cell types, I'm able to create differential expression gene lists for the clusters and compare them. These results can then be fed into a pathway analysis to get a better understanding of the cellular pathways that are altered by the condition of interest. Using this approach for the first time ever, we have been able to successfully characterize the gene expression profile of CD4 T cells at the site of infection, allowing us to identify different CD4 T cell subpopulations in their differential gene expression. This allows us to delve deeper into how helminth infection impacts CD4 T cell gene expression and the effect of Notch signaling in basophils for promoting T cell function during infection on a cell-by-cell basis. In the leftmost image, we see all of the cells collected from the mice that have passed quality control, which in this case is cells that call at least 500 reads associated with each cell, less than 7.5% mitochondrial genes present in those cells as they can be a sign of cell death. All of these cells are then cell scored to account for differences in cells due to the cell stage that they may be in. They are then clustered on a UMAP to show different clusters present in the sample and their relation to one another. I then created differential expression lists of the different cell types to find genes that are enriched in that cell type when compared to all other cells. Then I compared these expression lists to pre-existing cell marker databases and identify the cell types in my sample. This is seen in the middle and rightmost images that highlight the 10 most differentially expressed genes per cell type that are key in determining the cell type in question. This allows us to not only analyze the sample's response to infection and Notch manipulation, but to also look at how specific cell types and their responses to infection. The next step will be to analyze the T cell receptors that allow the cell to see or identify specific proteins or parts of the pathogen. The interest in looking at the TCR sequences is that it is currently unknown what TCRs are important in helminth infection. Preliminary work has been done with the corresponding T cell receptors as seen on the right-hand side to analyze how infection and basophil manipulation affects TCR diversity. The next steps will be to merge the TCR data with the existing clustered transcriptomics data to then see how the TCRs are responding to infection and basophil manipulation in different cell types and how they compare to one another. A better understanding of the response to helminth infection and manipulation of Notch signaling dependent cellular responses can lead to the discovery of novel treatments for helminth infection and allergic diseases, such as the development of better therapeutics and targeted vaccines. Lastly, I would really like to thank and acknowledge all of these individuals that I've worked with and have really supported me, especially Dr. Eliate Bueno, Dr. Lauren Webb, Jordan Cahoon, Dr. Bola Oyasola, and Hannah DeBerg at the Ben Arroyo Research Institute for all of their support.